Okay, so let's start. Yesterday we'd gotten as far as laying out the cross-sectional structure of the spinal cord. And we'd gotten as far as laying out the cross-sectional structure of the spinal cord and showing the gray matter and the white matter, and the dorsal root ganglia and the ventral horn. That's about as far as we got. So what I'll do today is I'll start with a discussion of using the slide of where we were yesterday. So I'll show that anatomy in a little bit more detail than we had on the board. Shouldn't be so hard. So we'll show that anatomy, and then we're going to move on and put into that structure. We're going to move on and put into that structure something known as the tendon tap reflex. The tendon tap reflex is one we're all familiar with. When you go to the physician's office and they check your reflexes and they put you sit down on the gurney and they come out with that rubber mallet and they whack you on the knee. That's the tendon tap reflex, and we'll walk our way through that cellularly and talk about what's happening at each of those steps. And as we walk our way through it, we'll see that basically it's a two-cell system. Unlike the system I showed earlier in the spinal cord, which was a three-cell system, a primary sensory afferent to an interneuron to a motor neuron. In this case, we have a primary sensory afferent synapsing directly onto a motor neuron. It's a very fast system. This motor neuron then innervates muscle. And that's the reason we get that twitch in your lower limb when we whack that patellar tendon. And we'll walk our way through that. But there's an additional idea I want to layer in on top of this, and this is known as reciprocal inhibition. And the reciprocal inhibition tells us that when we activate the flexor, that is the, try it again. When we activate the extensor, which is the quadriceps in the lower limb, simultaneously we inhibit the flexor, which is the hamstring muscle. And so we'll see that that pathway is a fairly simple pathway where the primary sensory afferent synapses on to the interneuron, which is inhibitory, which inhibits the lower motor neurons going out to the hamstrings muscle. So as we simultaneously activate the quadriceps, we're inhibiting the hamstring. And we'll walk our way through that circuit. This gives us some idea of circuits and how reflexes work in the spinal cord. But we're going to then carry that on to deliver this information, particularly about fine touch and proprioception, to the central nervous system. To deliver that information from the peripheral nervous system, to the central nervous system, we have two major pathways. The first pathway is going to be called the posterior or dorsal column medial lemniscal pathway. Now hold on to this idea. The medial lemniscal pathway, the PCML, the dorsal column medial lemniscal pathway, carries information about fine touch and proprioception. Proprioception tells us where we are in space. Fine touch is that delicate touch we talked about earlier. In both cases, these start out with stretch-activated channels. And I'll mention a little bit about those as we come to them. The other pathway is the spinothalamic tract. The spinothalamic tract carries information about pain, pressure, and temperature. Now there's terminology in here that I want you to hold on to, because it's important if you take any other neuroanatomy course. And that is when you look at spinothalamic. There's meaning in that word. Spino tells you where the input regions of the neuron are, that is the cell body predominantly. And the thalamus tells you where the output region is. So this will tell you that for this neuron in the spinothalamic tract, we'll see it's going to be the second order neuron. It has its cell body in the spinal cord and has an output region in the thalamus. And that pathway is going to be carrying information about pain, pressure, and temperature. Now we'll see that these two pathways are carrying slightly different kinds of information. But there are some things about these pathways that are similar and some that are different. And if we look at what's true for both pathways, is true for these sensory pathways, there's a primary sensory afferent, there's a second order neuron, and there's a third order neuron. That means that there's a three-cell pathway that's carrying information to your somatosensory cortex up in your brain. And those 3 cells are primary sensory afferent order neuron. Now, there's some rules in both systems which are true for both systems. That is, the primary sensory afferent never crosses the midline. So it has its cell bodies in the dorsal root ganglia, the posterior root ganglia. Those processes enter the cord. They never, ever cross the midline. They always stay on the same side of the body where they enter the cord. The next story that's true for both pathways is the second order neuron always crosses the midline. Doesn't matter whether we're talking about the spinal thalamic tract or the dorsal column medial lemniscal system. The second order neuron will always cross the midline. The third order neuron, in all cases, will receive input from the second order neuron. The third order neuron, in all cases, is found in the thalamus, in a place called the thalamic relay nuclei. So the first order neuron synapses on the second order neuron. Second order neuron synapses on the third order neuron. The third order neuron now will in turn send processes to the somatosensory cortex where it forms synapses. And we'll talk about all of this. And I hope today to get to the point where we talk about something known as Brown-Séquard. What Brown-Séquard is a hemisection of the spinal cord. And if we can walk through it and you gain understanding from it, you pretty much understand those systems. But let's start with this figure. This is pretty much where we left off. It's a much pretty picture that I drew on the board. It gives you much more sense of the three-dimensional organization of the spinal cord. But all the things we talked about yesterday and that simple line drawing are true here. So this is the cross-section of the spinal cord. Remember that in the spinal cord, divided the regions into the gray matter, which is that butterfly-shaped structure. And that's where the neural input regions were. Then outside those neural input regions, outside the gray matter, was the white matter. And the white matter were the myelinated axons sending information over long distances. Then we said that although this is considered to be all central nervous system, there's an interface between the peripheral nervous system and the central nervous system. And you'll remember from our very first lectures that when we talk about the peripheral nervous system, we're talking about sensory afferents and motor efferents. Sensory afferents carrying information toward the central nervous system, and motor efferents carrying information away. Well, that's recapitulated in this figure. The primary sensory afferent neurons have their cell bodies here in the dorsal or posterior root ganglia. The spinal cord is bilaterally symmetrical. So what you see on the right side, in this case it's showing left, and what you see on the right. Then this pattern is repeated along the length of the spinal cord. In fact, there are going to be 31 pairs of spinal nerves, all organized with this basic architecture, where there's the dorsal or posterior root ganglion that has all the neuronal cell bodies. We're going to see that they are going to send processes into the central nervous system. That's all going to be the dorsal side. So an important concept is when you see sensory, almost always think dorsal side of the cord. So the dorsal side of the cord is almost always sensory. And the ventral side of the cord, or the anterior side of the cord, is almost always motor. And so in looking at this is the dorsal root ganglion. This is the ventral root. Now remember, when we talk about roots, that's in the periphery. If we come into the central nervous system here in the ventral horn, or anterior horn, that's where the cell bodies of the lower motor neurons are. That's where we finished yesterday. And those lower motor neurons now will send processes out from the ventral horn, out into the periphery, through the ventral root, and then in the spinal nerve out here, we'll see the sensory processes and the motor processes will join in one nerve. I'll need to write it down, but write this down right now. A nerve is a collection of neuronal conductal processes in the periphery. We don't use that colloquially. Everyday language doesn't use nerve in that term, but in biology, nerve has a meaning. And the meaning is to be a collection of neuronal cell processes, conductal regions, if you will, in the periphery. In the central nervous system, we'll see that's called a tract. And that will bring us back around to the idea of the spinothalamic tract. So that's basically the overall structure of the spinal cord. And what I'm going to do now is I'm going to draw on the board the organization of something known as the tendon tap reflex. And the tendon tap reflex is one that we're all familiar with. And it's worth reminding you that if you go to the doctor's office and they want to test your reflexes, what they'll do is they'll have you sit in the end of the gurney, and then they come out with this sort of rubber hammer, sort of triangularly shaped, and they ask you to fold one leg over the other, and then the PA or the physician will feel around for this soft spot right below your kneecap, right below the patella. What they're searching for is something known as the patellar tendon. The patellar tendon attaches the patella to the bones of the lower limb. And that patella is also attached to the quadriceps, which is the major muscle of your anterior aspect of your thigh. So there's a tendon that goes through there, and they're searching for the tendon, and they take that hammer and they whack you in the knee. You know what's going to happen. They whack you in your knee, and your leg kicks up. The reason that your leg is kicking up is that you're contracting the quadriceps muscle. And the quadriceps muscle is an extensor. So when you extend your lower limb, you're pushing up your foot. So what we're doing is we're activating the quadriceps muscle. It's a very fast reflex. And it's a reflex that's important to us in terms of telling where we are in space. This reflex is important to us when, for example, we're walking down the sidewalk and we trip over a crack in the sidewalk. And we look around and hope no one's looking, but in fact, you'll very quickly recover, so that when you're walking along and you catch your toe, you very quickly will put weight on your left leg and hold up for a minute and then quickly put your foot out in front of you. That's a reflex. And that reflex is largely, not entirely, but largely dependent upon the tendon tap reflex and other stretch receptors throughout your leg and tendons that we're going to briefly summarize here as the patellar tendon tap reflex. So we'll start out here with one of my sorry representations of a leg. It's a terrible foot even for me. And in here, in blue, is the kneecap, your patella. Up here in red is the quadriceps. That's the quadriceps muscle. The quadriceps muscle is connected directly by a tendon to the patellar tendon. The patellar tendon directly, so the patellar tendon connects the quadriceps to the patella, and then down here to the bones of the lower limb. And this is the site that the doctor's looking for right here. It's the patellar tendon. Now, if we look more closely here at the muscle, so this is meant to represent the quadriceps muscle. Within the muscle, there are sensory nerve endings. So this is going to be the input region of a stretch receptor. Primary sensory afferent. It's the same story we told the other day. If we deform the end of this primary sensory afferent, if we pull on it, we're going to open stretch activated channels. As an aside, it was just this week that the Nobel Prize was awarded for understanding the stretch-activated channels, and they're given a name called PIEZO, piezo receptors. Turns out there are two of them. This is the type 2. The type 1 is found for blood pressure, but the story is the same in that when we pull on the membrane, we open these channels. What this receptor is doing is it's moderating, or it's modifying, or it's looking at the amount of stretch placed on the muscle. If we increase the stretch of the muscle, we open these channels. If we decrease the stretch of the muscle, those channels tend to close. So this is a stretch receptor in the muscle. It's monitoring the degree of stretch on the muscle. The story here is the same as we told the other day, where the stretch-activated channels, lead and sodium, brings the membrane to threshold of the trigger zone, fires an action potential. But let's follow that neuron into the spinal cord. So there's the spinal cord, there's the central canal. As we described yesterday, out here will be the posterior root ganglion, sometimes known as the dorsal root ganglion. And it contains all these neuronal cell bodies. Each ganglion may contain a few hundred to several thousand of these individual cell bodies for individual primary sensory afferents. Now, if we're going to follow one of these, we're going to follow one that we described here in the quadriceps muscle. So we can imagine here that this is the ending for the primary sensory afferent. It sends a process in the spinal nerve we'll eventually get into the spinal nerve, all the way back to where the cell body is located here in the dorsal root ganglion. Then the dorsal root ganglion neuron sends a process into the central nervous system. Unlike the story we told last week, this primary sensory afferent synapses directly onto a lower motor neuron. So this red dot is meant to represent a lower motor neuron. The lower motor neuron sends a process out through this structure here, which we call the ventral or anterior root. Remember, in the central nervous system, this region is going to be called the ventral or anterior horn. Horns are CNS, roots are PNS. Process goes out into the periphery, joins the spinal nerve, goes all the way down to the quadriceps muscle. And in this case, we have this lower motor neuron coming to the quadriceps muscle and innervating muscle fibers. This is a muscle fiber. So let's follow the pathway. We whack the patellar tendon. When we hit the patellar tendon, we move the patella, which pulls on the tendon attached to the quadriceps muscle. When we do that, we stretch the stretch receptors in the quadriceps muscle. That sets up action potentials that propagate all the way into the central nervous system, and then down here, at this synapse between the primary sensory afferent and the lower motor neuron, the neurotransmitter is the predominant neurotransmitter and throughout the central nervous system. This is going to be glutamate. The story then is the same as we've told so many times before. The glutamate is released. It binds to ligand-gated ionotropic channels. There's glutamate receptors. They allow sodium to come in to set up an excitatory postsynaptic potential. Now, this input is so strong that almost always you release so much neurotransmitter that you get a large enough excitatory postsynaptic potential, or a series of them, that you'll fire an action potential in the lower motor neuron. That lower motor neuron action potential now propagates along the conductor region of the lower motor neuron out to the muscle, innervating the muscle, telling the muscle to contract. So that's why when the doctor whacks your patellar tendon, your leg kicks up in the air because you have activated that extensor. But when we look carefully at the way our muscles are arranged, we find that for each one of these extensors, there's a corresponding muscle down here, which in this case is the hamstring. And this hamstring is known as a flexor. You know this more or less intuitively. You know that if you're standing on one foot and you pull your heel up to your rump, what you're doing is you're activating the hamstring. That's the flexor. And what we just described is how by activating the quadriceps, you'll kick your foot out. So that's quadriceps, that's hamstrings. So what we have here is, in the first part of the story, is activating the stretch receptors in the muscle, setting up action potentials in the primary sensory afferent, releasing glutamate onto the lower motor neuron, lower motor neuron reaching threshold and fire an action potential or series of action potentials to cause the muscle to contract. But the system is wired in such a way that we really don't want to simultaneously activate the extensor and the flexor. So we have a tiny circuit in the spinal cord. The tiny circuit is such that there's a branch of the primary sensory afferent. The branch of the primary sensory afferent synapses directly onto this orange neuron, and I'm going to label the orange neuron as an inhibitory interneuron. Well, the primary sensory afferent, again here, releases glutamate. The glutamate binds to receptors in the inhibitory interneuron. That glutamate is excitatory. It sets up EPSPs in the inhibitory interneuron. Those EPSPs are converted into an action potential at the trigger zone, propagate to the terminal, and now down here, we're in the spinal cord, so we're probably going to be releasing glycine. But the story would be the same if we were someplace else in the central nervous system and we would release the other neurotransmitter, GABA. Well, this synapse, this orange neuron, now synapses onto a lower motor neuron here. This lower motor neuron now goes out and innervates the hamstring muscle. So the way inhibition works in vertebrates is that all inhibition in vertebrates is occurring in the central nervous system. We don't have any inhibition out here in the peripheral nervous system. It's all in the central nervous system. And so what happens is, in this case where we excite the primary sensory afferent to release excitatory neurotransmitter onto the motor neuron that goes to the quadriceps muscle, we simultaneously excite the inhibitor, which releases inhibitory neurons. Oh, I'm sorry, which releases inhibitory neurotransmitter that inhibits this lower motor neuron. So we're now working this sort of yin and yang. When we activate the quadriceps, we're inhibiting hamstrings. But that inhibition is all occurring through interneurons here in the ventral horn. This same circuit exists. You could write the same circuit for a stretch receptor in the hamstrings. It would do if you could find the right place to pull on the tendon, it would do the same thing. It would activate those primary sensory afferents that propagate into the same cord. It would then synapse onto the lower motor neuron, go to the hamstrings, and that primary sensory afferent that was innervating directly the lower motor neuron going to the hamstrings would release neurotransmitter onto an interneuron that would inhibit the motor neuron going to the. So we're always running these things opposite one another. Yes?
If you did that, would then the hamstring be the extensor and the quadricep be the flexor?
So all we're saying here is that this muscle was always an extensor, because that's what it always does. this is always the flexor, because it's always what it does. And all we're saying here is that we can, if we excite the quadriceps, we will simultaneously inhibit the hamstrings. But if we want to flex that muscle, if we want to move our heel toward our rump, then we're going to activate the flexor and inhibit the extensor. But those muscles always do the same job. The only way we change them is by the neurons we activate. Okay? Anything else? So that's fundamentally the tendon tap reflex. And all of this is true. You're using these all the time. But if that's all we had, we wouldn't have any information reaching the brain. And we wouldn't really be aware of any of this. Our awareness of where our bodies are in space, our awareness when we touch something, all of that comes through projections of these primary sensory afferents and their synapses going to a region of the brain called the somatosensory cortex. And that's where we're headed now. What we want to do is not think about just the spinal cord reflex, which is really important, but in addition, how these neurons are going to communicate to other neurons in the brain. And that then we'll describe here. Take this panel, and we'll describe this as pathways to the brain. For somatosensory information. Now, when we talk about somatosensory information, we're talking about a handful of modalities. So this term, somatosensory information, comes in two groupings. One group is going to be said to be fine touch and proprioception. Both of these start out with those stretch-activated channels. But there's another type of modality, and this is going to include things like temperature and pressure and pain. By the way, the Nobel Prize that was awarded this week was shared by the fellow who discovered the stretch-activated channels with another guy who discovered the heat-sensitive channels by using the active ingredient pepper called capsaicin. So temperature is in fact activated by capsaicin. We also know that temperature for heat is activated by capsaicin, but you can also hijack the system using menthol to make it feel cool. But so this is going to be the pathway for temperature, pressure, and pain. This is going to be the pathway for fine touch, proprioception, and proprioception. So let's draw the spinal cord. And we're first going to follow a pathway known as the posterior or dorsal column medial lemniscus. Now that's a mouthful, so typically we'll abbreviate this as the PCML. In some cases you'll see it referred to as the DCML. Proximal, posterior column medial lemniscus. Well, let's look at that in the spinal cord. And remember, as I draw through this, I'm going to show just one side, but we're bilaterally symmetrical. So what you see on one side, you see on the other. Out here, in blue, is the dorsal or posterior root ganglion. That's where we have neural cell bodies. Now, all the neural cell bodies for both of these types of sensory information, fine touch and proprioception, temperature and pressure, all of those neuronal cell bodies are located in the dorsal root ganglion, either on one side or the other. The neuron we talked about, which was proprioception, came in and synapsed onto the lower motor neuron. But that's not what we're interested in now. This very same neuron, in addition to sending a process down to the ventral horn, will send a process here into a region known as the dorsal or posterior columns. You remember when we drew this yesterday, when we put the dotted lines on, and we divided the spinal cord into these quadrants, we said that up here was white matter. So these dorsal columns are going to be white matter. So they're going to be heavily myelinated. Now it's true that in the periphery, these primary sensory afferents are going to be myelinated by Schwann cells. But once they get into the central nervous system, they're going to be myelinated by the oligodendrocytes. And all along the process here, in the dorsal columns, we'll see this is white matter because those axons are myelinated. Now that's the cross-sectional view. Now I'm going to draw this slightly differently over here. And sometimes this is referred to as the ice cream cone diagram for obvious reasons. And what I'm trying to display here is that this region, the cone, if you will, this ice cream cone diagram, is the spinal cord. And I won't draw all of them in, but there are 31 pairs of spinal nerves. Let's follow one of them. Let's do our drawing of the leg again. We'll take the quadriceps muscle. We'll take the primary sensory afferent that we just described for the tendon tap reflex and bring this into the spinal cord. So here's the dorsal root ganglion. Sends a process in and it enters the cord. So this is what we've just drawn in cross-section, but now we want to follow this process. It enters the dorsal column system on the same side of the cord that it entered. Again, this is one of the rules that the primary sensory afferent, first it has the cell bodies in the dorsal root ganglion, and it never crosses the midline. That is, if it entered the cord from the right side, it always stays on the right side. If it entered the cord from the left side, coming from your left leg, it stays on the left side. So if we were to get on this neuron way, way out here in the muscle, we could walk along this neuron all the way back in to the dorsal root ganglion. We could hike across the cell body, walk on into the interface between the peripheral nervous system and the central nervous system, come down here and we'd reach a branch, a fork in the road. That fork would either go to the ventral horn where it will innervate lower motor neurons, or the fork could go to the right in this case, and we could now enter the dorsal columns. And in the dorsal columns now, we could walk along that axon all the way up into this region of the brain, known as the brainstem. Now, the brainstem is immediately above the first cervical vertebrae. That's right inside your skull. And we find that if we follow these pathways, we follow these neurons, they extend into the brainstem, and then in the brainstem, there's a region that I'll show in green. And this region is known as the dorsal, of course, also posterior column nuclei. Now again, there's some terminology here. The definition here is that when we talk about a nucleus, we mean a cluster of neuronal cell bodies. You can read input regions in the central nervous system. So you notice this nomenclature. When we were out here in the periphery, we were talking about ganglia. So a clustering or a grouping of neuronal cell bodies in the periphery is a ganglion, or plural is ganglia. But once we begin clustering these structures in the central nervous system, the nomenclature says that these clusters now get a new name. They're going to be called a nucleus. Just as when we begin to bring these neurons into the central nervous system, these bundles are going to get a new tract. We don't quite use that name for the dorsal columns. We'll see in a moment that once we get a bundle of axons or conductive regions in the central nervous system, that'll be a tract. In the peripheral nervous system, it's a nerve. But what we've done so far is we followed the primary sensory afferent into the central nervous system. It's entered the dorsal columns, sends a process up the dorsal columns, and then here in the dorsal column nuclei, that blue primary sensory afferent synapses onto a second-order neuron. And the second-order neuron crosses the midline. So this primary sensory afferent, whose cell body is in the dorsal ganglion and never crosses the midline, this primary sensory afferent synapses onto a second order that always crosses the midline. So again, I'll remind you that we're bilaterally symmetrical, so what we see on the right side we see on the left, so we can draw a midline in here. The second order neuron crosses the midline and joins a structure up here called the medial lemniscus. Lemniscus means a bundle of fibers. It's just a bundle of fibers. It's going to be this bundle of fibers that crosses over the midline in the brainstem, and ascends the cord a distance. I'm running out of room. And up here, in a region of the brain called the thalamus. In the thalamus, second-order neurons synapse onto third order neurons. I'm going to erase some of what I have up here because we need to find our way to the brain. So in the thalamus, the second order neurons synapse onto the input regions or cell bodies of third order neurons. This is a third order. And the third order neurons now will synapse onto neurons in the somatosensory cortex. So that's how we deliver information to the brain. And we'll deliver information to the brain about fine touch, discriminative touch, and proprioception through this pathway. So let me spend a minute here talking about what proprioception is, because it's really an important sense. Most don't recognize it in our everyday lives. But proprioception, because we're constantly monitoring the amount of stretch in all of our muscles and all our tendons, tells us where our body is in space. This is the reason you can close your eyes, bring your hand around, and touch your nose. You don't have to see where your nose is. You can have a coffee cup sitting on the table, And you can close your eyes and pretty much reach out and find it because you have this image of where your body is in space. You have an image where that coffee cup is, and you can reach out and find it. That's all proprioception. And this idea of proprioception, let me come back up to here. The proprioception all begins with the opening of the stretch-activated channels. It's true that when you're touching something lightly, that's also going to be fine or discriminative touch. That's proprioception. So another example of this is when you get home late at night and you realize that the lights are not on, and you realize you left your keys in your backpack and you have to get into the house. You know you can find your keys. You know your keys are in the backpack. You can open your backpack and you can fish around in the backpack until you find your keys. Then once you find your keys, you can work through each of the keys and you can find which one's for your car, which one's for the house, which one's for your bicycle, which one's for a door at work. And you can sort all those out just by touching them. That's all proprioception. We don't give this sense much thought. We're using it all the time. But yet it's really important to us because in that case, you could now find the right key, put it in the lock, which you found simply by touching the door, open the door, go in the house. All that done by proprioception. These fine touch and proprioception indicators all begin with stretch-activated channels out here in the periphery, and then they propagate information into the central nervous system. And through this pathway that we just described, all the way into the brain. But it takes 3 cells to get to the brain. Primary sensory afferent never crosses over. Second order neuron always crosses over. And third order neuron that extends into the cortex of the brain. Now that's the story for proprioception. Let me now take the other story for temperature, pressure, and pain. It's a very similar story. 3 neuron chain. Second order neuron always crosses over. First order neuron never crosses over. Let's put this in the back board. And now make the same drawing. Here's our foot. Now, rather than focusing on proprioception or fine touch, which would either be embedded in the muscle or on the skin, let's imagine down here, I'll put it in blue again. There's a neuron at the tip of your toe, and this is a pain neuron. This is the primary sensory afferent. As before, it has a process that goes all the way and joins the spinal cord. First, let me draw this as a cross-section. Here's the cross-section, here's the gray matter. This is the dorsal root ganglion. It has a neuronal cell body here. This cell body will be connected to this pain neuron. I may as well just draw it in. This neuron comes into the spinal cord. Now, immediately after entering the spinal cord, it forms a synapse. So it forms a synapse in this region called the dorsal horn. Well, it's always 1, 2, 3, so this primary sensory afferent, I'm telling you it's a synapse here, it synapses onto the second order neuron. The second order neuron crosses the midline, goes to the other side of the cord. So here we are with the midline. This second order neuron now enters a region of the spinal cord over here known as the spinothalamic tract. So here are the rules. Primary sensory afferent never crosses over. Second order neuron always crosses over. Now, where in the DCML, the second order neuron crossed over in the brainstem, here we're crossing over in the spinal cord. If you take a course in neuroanatomy, you'll find that the brainstem is largely an extension of the spinal cord. But in this case, we see the primary sensory afferent onto the second order neuron, joining the structure known as the spinothalamic tract. Again, there's meaning here, and I'll hold you to this meaning. This is where the location of the input. You can read cell body or dendrite. This next part, thalamic, tells you where the output region is. And this is nomenclature we use throughout the nervous system. And if we follow this pathway over here, so what we're going to do here is we're going to draw the ice cream cone diagram again. See ice cream cone diagram? Let me make that a little smaller. Spinal nerves. This is the dorsal root ganglion, or posterior root ganglion. The neuronal cell body will be located here. It will enter the cord, and almost immediately after entering the cord, it synapses onto that green second-order neuron. The second-order neuron then crosses the midline and ascends the cord in this structure known as the spinothalamic tract. The spinothalamic tract now is carrying the axons, the conductor regions of the second-order neuron. Spinothalamic, we know from that nomenclature where the synapse is going to be. The synapse, again, is going to be found up here. Hope I labeled it the other one, called the thalamic relay nucleus. So just as up here, we had this thalamic relay nucleus. Again, I have to finish. The third-order neuron will receive input in the thalamic relay nucleus, send a process here to the somatosensory system. Somatosensory cortex. So again, let me quickly summarize this. In all cases, the dorsal root ganglion contains the cell bodies of the primary sensory afferents. Primary sensory afferents never cross the midline. All primary sensory afferents synapse onto second-order neurons. Second-order neurons in both the PML and the spinothalamic tract cross the midline and ascend to the thalamus. And in the thalamus, the second-order neurons synapse onto third-order neurons. Third-order neurons synapse onto cells in the somatosensory cortex. And what I'm going to leave you with is what happens if we cut the cord halfway, we hemisection. If we hemisection the cord, what information will get through and what information we blocked. I'll leave you with that, and that's where we'll start tomorrow.